I want to thank the organizers and also thank all of you for showing up early in the morning. Trefoil is developing breakthrough therapies for corneal disease. The key points that I'd like to leave you with today are first, that persistent corneal edema is a large and unaddressed market. It is one of the most common reasons patients are unhappy with their cataract surgery, and it affects about 400,000 patients per year in the United States. The pathophysiology driving edema, both in post-surgical edema and in Fuchs' disease, is well understood, and is directly impacted by the pharmacology of our lead drug, TTHX-1114. And we have clinical data that show that TTHX-1114 accelerates the resolution of edema and improves vision, and I'll show you some of that data. We forecast this at a peak sales of about $800 million a year in the United States alone, and we're currently raising a Series B to try and drive this clinical program forward. So corneal edema has a significant impact on vision, and this just illustrates what it looks like to a patient, but quantitatively on the right-hand side, you can see that there's a quantitative relationship between edema and poor vision. Almost all patients with severe edema, most patients with moderate edema, and even many patients with mild edema don't see very well. And we use 20/40 as a benchmark. They see more poorly than 20/40, but that's an important benchmark because that's how well you have to see to drive in most states, and that has a significant impact on the quality of life. The population here is mostly patients that have undergone some form of ocular surgery, in particular cataract surgery. The risk factors for post-surgical edema and corneal decompensation are well recognized, but there are still patients that don't have risk factors, that have edema. And of course, there are non-surgical patients, such as Fuchs patients, that simply have progressing edema as a consequence of their progressing Fuchs disease. Our approach to this is our lead compound, TTHX-1114. It is a biological product that is a modified FGF1. It has the same inherent properties, interacts with the same receptors as native FGF1, but we've made some molecular changes to increase its stability and prolong its half-life in tissue. What's really important about this is that it directly reverses a molecular process called ferroptosis, which we know to be a driving factor in corneal edema in corneal endothelial cells. It has high potency, we have strong intellectual property, and because it's an easy-to-make small molecule, it has very low cost of goods. The molecular and cellular biology of this process is well understood. The endothelial cells that line the inside of the cornea are responsible for two functions. The first is a barrier function to prevent fluid from leaking into the cornea, and the second is a pump function to pump fluid that has leaked into the cornea back out. Two functions are both compromised by oxidative stress. Vacoemulsification is a direct application of oxidative stress to the endothelial cells. And it's sustained by this process called ferroptosis. What happens first is that the barrier function breaks down, and second, the cells become unable to make ATP, so they can't pump. That whole process, which again is molecularly driven by a process called ferroptosis, which is a positive feedback loop, is actually blocked by our drug. In order to prove this in humans, we turned to a population of patients that were undergoing a procedure called decimate stripping only. These are Fuchs patients. And the procedure always induces severe edema. And in that study, which is the STORM study in the middle here, we had 49 subjects. We've done 3 clinical trials, including that study, and demonstrated first, before I talk about efficacy, that the drug is safe and well tolerated. We've administered it to a total of 112 subjects, 355 drug injections. There are no dose-limiting toxicities and no drug-related significant adverse events, so it appears to be well tolerated. In the study of patients with DSO, we looked to see if we could accelerate the recovery, the resolution of edema, and most importantly, the recovery of vision. And this slide shows that there is a dose-dependent and statistically significant recovery of vision. We have most of these patients recovering good vision after 21 to 28 days in a situation where untreated, these patients would be expected to take 2 or 3 months to recover good vision. And so we have strong evidence from this human study that the drug is active. So the status to date is that we have demonstrated a mechanism of action. We've demonstrated that the drug is safe. We've demonstrated activity in that DSO study, and we are now moving on to a large, well-controlled, randomized mass study called ACE to demonstrate that it works directly in the patient population of patients that have edema, whether they have post-surgical edema or edema secondary to Fuchs disease. We do have another program for a topical formulation of the same drug directed at corneal epithelial conditions with the common factor of epithelial compromise with inflammatory components, because we know this molecule is immunomodulatory, and we have completed a phase 1 safety study and are ready to go into phase 2. And with that, I will thank you and remind you that we are in the process of raising a Series B, and I'll answer questions at the break. Thanks.